Hello, and we're going to be getting into a chapter now where we're going to be revisiting evolution. Now that we have learned about genetics and biochemistry, and we'll be learning a little bit more about the origin of life on Earth. So we have to really start to where it all began, and that is with the formation of Earth. So the Earth, based on not carbon, but radioactive dating methods that we have, the Earth would have formed 4.5 billion years ago. The entire process took about 100 million years. And after all of these collisions of cosmic dust and debris, and with everything melting together, and all of this material condensing and rearranging, the most dense material would be at the core, and then the less dense material above. So we would have our Earth, right? So it starts there. Our moon forming, that would also be quite a monumental event. That would have collided with Earth, and then that would get stuck in the Earth's rotation. Now, if we are looking at how life first formed, we need to think about what materials there would have been on Earth before that life would have first formed. So we know that there are different organic macromolecules. We know that there are proteins, that there are nucleic acids, that there are lipids, and that there are carbohydrates. Now, quite notably, but there are in all of those macromolecules are carbon, hydrogen, oxygen, nitrogen, right? Also sulfur and phosphorus. So if we look here, in the early Earth's atmosphere, we see different gases, right? And there's a whole list of them up here, and I'll stick to talking about the ones that are in this diagram here. But we see nitrogen gas, we see carbon dioxide, we see hydrogen gas, we see methane, and we see ammonia, we see NH3. But if you think about it, all the elements that we need for life, for the basic macromolecules that make up living things, are there. Whereas in Earth's modern atmosphere, probably the biggest difference that we have there is really the presence of oxygen. So as the Earth cooled, our first rocks would form around 4 billion years ago. Volcanic eruptions would continue, intense thunderstorms would continue, forming liquid water. So we have an idea as to what early Earth was like. And if we have an idea of what early Earth was like, an idea of what gases were in early Earth, and if we have this question as to what happened on early Earth, is there a way that we could simulate what happened on early Earth? And so that's what Miller and Urey would do back in the 50s. And this is how they set up their experiment. So we have a sealed tube system to simulate conditions on early Earth with everything sterilized, so no oxygen because there was no oxygen there. Electrodes to simulate lightning, and in the end, this is what they collected. So here's their experimental apparatus, and here at the first step, we have a bulb with water being heated, and that's supposed to simulate the volcanic eruptions that would be occurring, heating up the water. Here at step 2, there's a mixture of methane, which is CH4, ammonia, which is NH3, and hydrogen, which is H2, that's added to the water vapor. So these are gases that would be in the early Earth's atmosphere. Here you have step 3. The circulating gases are bombarded by sparks of electricity. So that's supposed to simulate the lightning that's in all the thunderstorms. And then step 4, here, there's cold water. So this is going to flow through something called a condenser that cools the chambers, causing droplets to form. And ultimately, here in step 5, after a week, liquid is cooled, which contains amino acids and other organic compounds. So we start out with some gases, CH4, NH3, H2. We start out with these gases that are in the atmosphere. And then what we end up with is the formation of some of these amino acids, which make up the building blocks of living things. One of the organic compounds that we talked about, right? Because proteins direct a number of life processes, and amino acids are the building blocks of those. So as we get more accurate information for life on Earth, these experiments would be performed more, and we would be able to form uracil and cytosine. And so it's evident that there are simple organic molecules that are able to form in this environment. So what is it that we need in order to form real cells? So basically, what's necessary for a cell? Well, the first thing that you need is a barrier to the outside environment. So you need a cell membrane. You need hereditary material, so some sort of genetic information, whether it's DNA or RNA, which came first, we'll discuss that, and a way to get energy. Now, in terms of cell membranes, how would those have formed? So a cell membrane is a collection of these smaller phospholipids. So in order for the first cell membrane to form, it might have been a collection of organic molecules, right, surrounded, that came together. So these early cells, we call these microspheres, collections of organic molecules surrounded by a membrane. And so it's fairly easy to form these in lab, and there are certain lifelike qualities that there are to these microspheres. I mean, one thing is they have some sense of organization, right? So they have materials that can be enclosed within a membrane. But it is also possible for another microsphere to kind of bud off of the other microsphere, and therefore they can increase in number in that way. Now, when we were talking about the things that are necessary for life, for cells, we were talking about the fact that there needs to be a barrier, a cell membrane. And here, so where did heredity originate? We can't say for sure, but if we talk about DNA versus RNA, there's a lot more evidence that suggests that RNA would have come first. So we have discovered ribozymes, which are just catalytic pieces of RNA, which can store info and catalyze reactions. So returning to RNA, we had inorganic matter, and first the assembly of just the individual monomers, the assembly of RNA nucleotides. So RNA nucleotides would have been able to assemble into a chain. And we know that RNA is able to replicate itself. It's able to synthesize proteins, and it can function in information storage. So it has those 3 capabilities. So that's what RNA is able to do itself. So how would RNA link up, or how would RNA become the DNA that then itself can also function in information storage. How would RNA become another type of RNA that helps in protein synthesis? Because we talked about how there's different types of RNA. And then what is that step that goes from the assembly of RNA to the first proteins that would be made? So there are some steps in this process that still have yet to be worked out further. But it's most likely that RNA would have been our first set of genetic material. Now, what exactly happened, we can't really say for sure, but we've tried to recreate conditions to create cells in the lab. But at some point, conditions were just right in order for the right mix of molecules to form. Now, what were the first cells like? So there are microfossils that have been discovered to show that there are prokaryotic cells about 3.5 billion years old. So there's something called the heterotroph hypothesis, which is that the first cells were probably heterotrophic prokaryotic cells. And your earliest cells probably also lived in the most extreme environments. They probably lived in deep sea hydrothermal vents. Your earliest eukaryotic cells would have formed as as different cells would engulf other cells. But let me use this picture to explain. So here in the origin of the eukaryotic cell, this larger cell here would have been an anaerobic prokaryotic cell. So it's a cell that is able to survive without oxygen. So that's what anaerobic means, right? We were talking about anaerobic respiration a couple months ago. So this cell, in order to get nutrition, would have to consume other cells, like this other bacterial cell. Except in this case, this cell is aerobic, so it's able to use oxygen. Now, this cell would normally consume and break down other cells, except in this case, this would be a primitive eukaryotic cell because this cell will have incorporated what would be our future mitochondria. So the reason why this cell or this future mitochondria didn't get digested was because this mitochondria produced a lot of energy for this larger host cell, and that would be like the first eukaryotic cell. So some of these cells would later on go on to consume other photosynthetic bacteria that would eventually be your future chloroplasts. And these cells would be able to provide their own oxygen for themselves. And that would be like your first plant cells, your first photosynthetic unicellular eukaryotic cells. So this is what we call the endosymbiotic hypothesis, because as the cells were taken in endo, they formed some sort of symbiosis, symbiotic, so a mutually beneficial relationship. So that's what that means. So this connects with the picture that I just showed you, because initially we have a whole environment with heterotrophic cells, but at some point photosynthetic bacteria would evolve. But our first photosynthetic cells, probably not exactly like today's photosynthesis. Today we have photosynthesis where it's water that donates the electrons. Today where it's H2O that donates the electrons. Initially, it was probably just H2S that donates the electrons. But with photosynthesis, it would change the atmosphere forever. So the formation of the Earth would be about 4.6 billion years ago. And then a billion years later would be our first cells. Then a billion years later would be the first photosynthetic cells. So that's a timeline of our history. So photosynthesis would have a huge impact on life forms that are present because up until that point, living things existed without oxygen. So all of those life forms that existed without oxygen died because oxygen was toxic to them. As more and more oxygen was produced, the oxygen would form an ozone layer. So this ozone layer would filter out a lot of the UV radiation that would come to hit our Earth. And that is beneficial because that prevents the UV rays from causing mutations in molecules, and aerobic life can flourish. Now, this I probably could have taken the picture I had on the other slides and then copy it and put it on this slide here, because those are the first eukaryotic cells. So imagine I put the picture here. But that is the endosymbiotic theory about 1.5 billion years ago as to how our first eukaryotic cells would have evolved. And with sexual reproduction, that leads to variation, and that would accelerate the rate of evolution. Because with sexual reproduction, we have a recombination of genetic information, and that is going to allow evolution to occur at a faster rate. So there are different views, right, as to how life would have originated. Panspermia is the idea of how life would have been brought to Earth from outer space. Could a meteor have carried cells? But there's really no evidence to suggest that this is true or would be viable that way. Your Ed puzzle gets into this. Now, what is spontaneous generation? It would ultimately be disproved by Louis Pasteur, but spontaneous generation was a prevailing belief at the time that living things came from non-living things. There's a number of things in science that go against people's beliefs, and they're things that we take for granted today, but it would take a long time for science and for scientists to convince people otherwise. I mean, it would take a while for science to convince people that, or to convince the public that the Earth revolved around the sun instead of the sun around the Earth. That took a while, right? It would take a while to convince people that the Earth is round and not that the Earth is flat, right? It took a while for people to understand the idea that there's microscopic life, that there's life that they cannot see. That took a while, right? So there's always these scientific ideas that seem to be very, very abstract that is hard for the public to grasp because they are abstract concepts. This is one of them that we take for granted today, but at some point back then, people really believed. Spontaneous generation is the idea that living things arise from non-living things. Biogenesis, life from life. So Francesco Redi wanted to disprove the idea that living things come from non-living things. He wanted to get rid of people believing that. People believed that if they took a piece of meat and put it in the back of their barn and it came back days later, that the maggots that they see on that meat came from the meat. They believed that. They believed in spontaneous generation. They believed that there was some vital force in the air that turns the meat into maggots. They believed that. So Francesco Redi said that that's impossible. He said meat cannot turn into maggots. This is the experiment he set up to disprove that. So here in his control group, he put meat in jars and he left it open. Here in this jar, he put meat and he covered it with lids. And in this jar, he put meat and he covered it with like netting. So air could get in, but the flies could not. So Francesco Redi says the maggots come from flies that lay microscopic eggs on the meat. You guys can't see the flies, but I'm sorry. You guys can't see the microscopic eggs, but the eggs are there. So sometime later, there are maggots on the meat that was exposed, but there's no maggots in these two groups. So obviously, the flies got in here, but the flies cannot get into this group here or this group here. Now, the people who believed in spontaneous generation believed that there was a force in the air that could turn meat into maggots. So they would have said that in this group here, of course, the meat didn't turn into maggots because the vital force couldn't get in. But they were left speechless. They couldn't defend themselves when they saw this experimental group here because the force in the air could still get in, but the force could still get in. So why is it that there were no maggots? And that's because the flies couldn't get in. So Francesco Redi's experiment in the 1600s was important with that. But we also know that there is microscopic life. So here's another experiment to try to get people on board with believing that life doesn't come from non-living things. All right, so that life doesn't just spontaneously come out of nowhere. So microscopes showed the presence of microscopic life. So Spallanzani, in the 1700s, argued life comes from life, and that, like, if you left out soup broth, it becomes cloudy because of microorganisms in the air falling in and growing. But his experiment was criticized by others because it locked out the vital force in the air. So here was this experiment. What he did was he had this Erlenmeyer flask. He heated it up to sterilize it. He left it open, and then it became cloudy because it was exposed to the air and the elements, and the microorganisms got in and caused it to become cloudy. Set up another group. Here, he sterilized it by heating it up. He had it corked, and there was no growth. But the people who believed in spontaneous generation disagreed with him because they said, of course, there's no growth because the vital force cannot enter. So they were not convinced otherwise. Along would come Louis Pasteur. This is where we get the word pasteurization from, thanks to this guy here. So this would happen in the late 1800s. So Pasteur would basically take Spallanzani's experiment. And what you see here is there is a experimental apparatus of broth, and what he does is he heats it up. He sterilizes it. And here's a curved neck flask, or like a swan neck, okay? So then he removes the heat because he's already sterilized it, and this swan neck traps microorganisms. So he could leave this alone for years, and there will be no microscopic growth occurring inside because the microorganisms are not able to crawl up the swan neck and get into the broth. But if you break off, so if you do the same experiment and you break off the swan neck, there are microbes that are able to get in and grow. So for someone who does believe in spontaneous generation, this would put them to be, this made them hard to defend their position because the air and the vital force are still able to get into the broth, but here the microorganisms would be trapped. So Louis Pasteur would ultimately disprove spontaneous generation because with this curved neck flask, yes, the vital force can enter, the microorganisms cannot, and we don't see the broth just turning into these microorganisms. So finally, after all these hundreds of years, spontaneous generation would be disproved. So, um, we, right, we've been talking about how our first we've been talking about the first cells and timeline of life on Earth. As we go forward, what we'll be getting into is we'll be talking about evidence for evolution. We'll be talking about how we can date fossils. We'll be talking about mass extinctions. So, um, so what I want to show you was this timeline of history for life on Earth. Now, um, the geologic timescale can be broken up into these major eons and eras and periods. So, this bottom portion here, which doesn't seem to be represented by much area here, but really does make up 90% of the Earth's history. So this is not completely drawn to scale here, is the Precambrian era. So really, Precambrian era here, this should probably be like 9 times as long. But it would be during this time that prokaryotic cells and eukaryotic cells would start to evolve, and all of our life would be in the seas, and there would not be many fossils. Following the Precambrian era, we have the Paleozoic era. Okay, so the Paleozoic era, which is this time span here that my arrow is over, is from 500 to 245 million years ago. But this is basically the age of fish. So our first vertebrates would appear, mostly jawless fish, and we would have our first land plants. So we would have photosynthetic organisms in the oceans, but then we would have land plants evolve. So that would allow for that would open up a bunch of niches on land that other life forms could then occupy, like amphibians. So vertebrates like amphibians would begin to invade land at the end. Now you see mass extinction. So a lot of these eras are denoted at the time that they are because they are defined by mass extinctions. They are defined by extinction events. So one of the things you guys are going to be looking at this week is the extinction events of the dinosaurs. So this is why I have to introduce this to you here as well. So the Mesozoic era, that would be the age of reptiles. And so that's from 245 to 65 million years ago. That's this time right here where my pointer is, the age of reptiles. The Jurassic and Cretaceous periods where dinosaurs would rule the earth, angiosperms would evolve. So those are your flowering plants, and very, very small land mammals. So again, at the end of this era, there would be another mass extinction. Again, so a mass extinction that marks the end of these eras, and that would wipe out more than half of the life on Earth. And so we have our age of fish, then we have our age of reptiles, and then we're in the current era, which is the Cenozoic era, and we have the age of mammals. So that's from 65 million years ago to the present time. So we have our first human-like ancestors called hominids at about 4.5 million years ago. And the first Homo sapiens would emerge around 100,000 years ago. And modern Homo sapiens around 50,000 years ago. So here's the Cenozoic era here. And you can read articles that we are arguably in a sixth and arguably in another mass extinction event right now caused by humans. So there's an alarming rate of species that are becoming extinct really due to our own influence. So here are mass extinctions occurring over time, and we have these major events which would mark the ends of major eras. And this is the one that would mark the end of the dinosaurs, the end of the Mesozoic era. And I'm going to have you guys be looking at something this week that goes this week into next week that goes a little bit deeper into that. But that's it.